Every year, 5 billion people are at risk of infection from malaria. Of that, 212 million are actually infected. And in 2015, 419,000 lost their lives to this disease. Most of them were under the age of 10, with a staggering association with poverty. My PhD aims to provide new material in this fight by looking at a molecule and atoms in the malaria parasite. We call this molecule kinesin. And if you want to understand a bit about kinesin, I'm going to take you through the story, but you have to see it in its social context, the hustling and bustling of the cell, the exchange and interaction that goes on in between. You have to see a molecular machine. And so a molecular machine is much like any other machine. It's a complex multi-component system that requires the input of energy to then achieve a mechanical function. My machine we call the Spindle Apparatus. It's a huge machine consisting of 600 different types of proteins alone, and it exists for one purpose, which is the separation of copied DNA. And this is no small means. When you think about a cell dividing, it needs to inherit genetic material, and so DNA is copied, and the Spindle Apparatus, it takes this DNA, spreading it to each side of the cell, so the cell can divide squarely down the middle. And so we continue on. But I really want to give you a kind of a more visual, more interactive view of the mitotic spindle. So we'll go in for a visual experiment. Close your eyes if you like, but imagine yourself in the middle of the cell. In front of you sits the huge nuclear membrane, but as soon as you're there, punctures start appearing in that membrane. It rips apart, revealing huge strings of DNA, masses of it, and they then start to condense, coiling into little neat packages. And so we then see our mitotic spindle, the spindle apparatus, and the kinesin molecules. It starts off small in the corner of the cell, and then it splits exactly into little fibers begin radiating out. You see kinesin with its long stalk and globular end feet starts to drag against the fibers, pushing it apart. The two halves split and move to the other side of the cell. By this point, the spindle apparatus is massive. All of its fibers have completely consumed the innards of the cell. Some of them begin to connect to the copies of DNA, forming little neat structures. The spindle apparatus then begins to jostle, aligning, waiting for the time to be right, and the cell sends the signal, and the spindle collapses, dragging the DNA to either side of the cell, so it can then split in the middle, and so on and so on. All this is intriguing and fascinating detail, but it's not really why we're here. There's another interesting property of machines that is what's driving this forward. Yeah. Thank you very much.